Good morning, everybody. Welcome to day 4 of BCBT. And today we are continuing the theme of olfaction. And we have, or in the first talk certainly, and in the second talk I think we're moving to a slightly new topic. But we're beginning with the talk from Bill Hanson of the Max Planck Institute for Chemical Ecology, who's going to talk about evolutionary neurology of the olfaction system in flies, and is going to tell us about good and bad odours and the correlates of valence in Drosophila. So this should be a really interesting talk, and over to you, Bill.
Thanks. Morning. As far as I get it, a lot of you guys are computational neuroscientists, consciousness, building robots, and so on. So I'm going to talk much more about nature today, but you all have to factor in nature when you're dealing with the nervous system, because that's what it evolved to deal with. So I'm going to talk about how good and bad is coded, more or less, and what is good and bad for, in this case, for a fly when it's trying to survive in the surroundings. So we have an immense literature during the last few years over what's been going on in the Drosophila olfactory system and how they code, what the receptors look like, what the brain looks like, how different, more or less artificial odors taken from the shelf of the lab are coded, are detected, and so on and so forth. What has been left out a lot is how the flies actually survives where it lives. So this is what the fly has to deal with, right? So it's moving through a pretty complex environment, and out there it has to find good stuff and avoid bad stuff. That's more or less the life, right? And then finding good stuff means you have to find food and you have to find somewhere to lay your eggs. Avoiding bad stuff is more or less avoiding eating bad stuff and avoiding being eaten or parasitized by some bad other animals. So that's more or less what we're going to deal with. You haven't looked at a fly. This is a fly from the front. Molecules are hitting the antenna, which is the main olfactory organ. It's a little bit glitchy here, but you see all these hairs on the antenna. Those are the olfactory organs. The molecule goes through those hairs, and inside the hair there's a lot of nerve endings. On these nerve endings there are the receptors. The molecule hits one of those and activates the nerve. Okay? So that's the nose of the fly. From there on, it's a little bit slow this morning, the computer. Come on, there we go. From there on, the message passes into the main olfactory center, which is the antennal lobe. The antennal lobe is built from little balls, just like in every other organism that has an olfactory system. So glomeruli, I'm sure you heard about these yesterday as well. These take care about the message from each neuron type. So each neuron expressing a certain kind of receptor will project into one of those little balls. So in this way, a three-dimensional topographic map is built in the antennal lobe of these flies. From the antennal lobe, the message is sent up to higher centers of the brain, like the mushroom body, which is the hippocampus of the fly brain. That's where a lot of the experience-dependent behavior is formed, and to the lateral horn, which is we think is more naive behavior, where you don't need to learn so much. So I will talk about the antenna a lot. I will talk about the antennal lobe, and I will talk about the lateral horn in the end of my talk. Okay, so just when I moved to Jena some years back, we decided to go back and look at what do all these odors that people have looked at really mean to the fly, because again, Drosophila has been used as a flying test tube, more or less. So it's people have given it odors, but at the same time they haven't really cared what do these odors mean to the fly. So there was a huge publication from John Carlson's lab some years back where they looked at 110 odors. How are these odors coded on the antenna of the fly? So we took these 110 odors and looked at what do they really mean to the fly. So blue here to the right means that something is good. Pink to the left means that it's bad. And then we went through all these odors in a choice assay where the fly could choose to go to this odor or to go away from this odor. And you will see here as we go down, 110, a lot of experiments, tens of thousands of flies, a lot of good odors, a lot of neutral odors, very few bad odors that the fly will avoid. The most, if you look farthest down there, benzaldehyde, that's the worst odor of all for the fly so far, which I'm going to show you something else. But what we then did after this, we took the 6 worst and the 6 best odors, and then we went into the brain and did imaging. This means that you express a calcium indicator in the nerves inside the brain, and then you image the antennal lobe while applying the odor to the antenna, and then you can observe the patterns that are formed in the antennal lobe. So the antennal lobe is these 2 parts here, and then you see the projections to the mushroom body and to the lateral horn of the brain. Okay, so then we did all this imaging of all these things, and what we could find, and this is very briefly telling, is that there is a coding of good and bad. There is a clustering of good and bad in the antennal lobe on the output, at the output level. So things come in pretty not so organized, but when it leaves the antennal lobe after being processed in there, we see a clear separation of a cluster of glomeruli coding bad stuff and a cluster of glomeruli coding good stuff. So this was sort of the start of our venture into good and bad, that we saw that there is really something to it. There is we can see more or less, by observing a pattern in the brain, we can see that this odor is probably good and this odor is probably bad. So that's the first part, and if you want to read more about it, it was published here in a couple of years back now, 1 year back. Okay, so then I want to move.
Is that what you mean by topography? You said there was topography in the antennal lobe.
Yeah.
Okay.
Functional topography, if you sort of, yeah. So now I want to go into the first story, and this is a published story. Everything after this story will be unpublished stuff. So this is all about a stop signal. So it's something that is really bad that we found out for the fly. It's something like when you smell a dead animal, or when you smell feces, or there are things that we are born to dislike, more or less, and we think that this is one of those odors that we discovered here, and it's all connected to the system that you see here on the screen. It's all about this molecule. This molecule is geosmin. Geosmin is a famous molecule because it's one of the compounds that we are most sensitive to as well, and it's actually the smell of moist soil. So if you go out in the spring and smell when the farmer is tilling his soil, this is the nice odor because it's low concentration and you have a positive feeling, right? But it's also the odor when you go into a very old basement and you smell and it's not so nice, right? It's the rotten, moldy smell. It's the smell of corked wine. When you get a red wine where the cork has molded, it's also geosmin. Okay. So geosmin is also produced by microorganisms. It's a well-known microorganismic odor, so from penicillium, from streptomyces, also from blue-green algae, but for us, these 2 here were pretty interesting. So what we started with was experiments up in my old lab in Sweden, which I'm also still involved in, was that we started flying flies in a wind tunnel. So we built a small wind tunnel and we tried to get them, because that's also something that people had never really cared about. They were watching the flies more or less moving in a petri dish. That was the whole thing. Maybe I should put my phone somewhere else. It's disturbing. So what we could establish there was very nice attraction to vinegar, right? But what we also could see when we added geosmin to the vinegar was that we abolished all attraction. So a little bit of geosmin, and this was all the idea of Paul Becher, who is the first author here, because he used to be a microbiologist. So he thought that could be a cool odor to test, one of these serendipitous events in science. So we just tested it, and we got a very nice effect. It abolished attraction to something that the fly really loves. It would be like you being extremely hungry, seeing a big steak, Heather, in front of you, and then suddenly you get a little whiff of a cucumber beside it. Say, no, that's not nice, right? So things like that, something that prevents you from going to a resource that is really good. So our question was, of course, that flies really love vinegar. They will really. You can test it in your kitchen. Put some balsamico out there. Put a drop of dishwashing detergent in there, and then in the morning you will find 20 flies in there, I'm sure. So why? Why do the flies not go to the balsamico when we add geosmin? So let's start dissecting this system now. We started from scratch, so we had to re-establish in our lab with our bioassays that geosmin is really repellent. So we chose this simple bioassay where the flies can choose to go or not to go towards an odor. And we established that geosmin is amazingly repellent. This is benzaldehyde, the most repellent odor ever known for Drosophila before. If we look at geosmin, you see 1, 2, 3 decadic steps more repellent. But that's not enough, because if you look at these two compounds, benzaldehyde is about 1,000 times more volatile. So it's 1,000 times 1,000. So it's 1,000,000 times. Geosmin is 1,000,000 times more efficient as an inhibitor of behavior for Drosophila. So it's something that we have not seen so far. It's almost active in homeopathic concentrations. Extremely negative. Okay, so then we went in, we have established a new behavioral paradigm. Behavior is very important for us because we're neuroethologists, so we always want to have good behavioral background to what we look at in the nervous system. So we went into our fly walk. Our fly walk is where Paul has also been heavily involved and his people to establishing a system that intelligently looks at all these flies running in 15 tubes, constantly observing them and seeing what each fly does when an odor puff hits it, right? And then, so this is more or less what it looks like. We add the odor, the flies are sitting in there, and one problem is, of course, that you cannot train flies to sit in a line. Each fly will sit somewhere else in each tube, so you have to calculate exactly with the speed of the wind and when the odor will hit the fly, and then the computer looks at them all the time and says, now it hit, and this is what the fly did. Then we get these kinds of diagrams, and you see here, when we only give the fly balsamico, then it runs very nicely upwind towards the odor. When we add geosmin to the balsamico, this is totally abolished. We get very nice activity in the fly when we add this, abolished with geosmin. So geosmin gives some kind of freezing behavior on its own. So it is something that the fly really reacts to. Well established now. So the flies are really, really sensitive to geosmin. So then our next question is, of course, how does the fly do this? What do they do to detect geosmin? First thing, where is it detected? Because I told you the antenna is the fly nose. That was sort of half the truth, because the fly has two noses, the antennas, but they also have the palps. Both of these are noses, and the fly detects different kinds of odors, as you will see later on in my talk as well. So we went in, did simple electrical recordings from the antenna, from the palps. We could show that it's detected on the antenna. Then we can knock out the co-receptor, which every olfactory receptor needs to function. We knocked that out, we lost the response. So we knew that geosmin is detected by an insect olfactory receptor. That was the result of these first experiments. Okay. So, which one? We have about 60 to choose among. So where do we start? We start on the antenna. Here is the antenna in a better picture. You see all the hairs there. Here is one of these hairs cut through. You see that the nerves come up and they branch in there. Then we go in with electrodes just at the base of these hairs, and we record what is happening in these nerves when they're hit by different odors. Then an extremely good Egyptian PhD student working in my lab did thousands of recordings. He recorded from every sensillum type on the antenna. You see them all there. AB1 is one sensillum type. It has 4 cells inside, A, B, C, D, no response. AB2 has AB, no response, blah, 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 blah. All of these were tested with geosmin. And as you see, one single type responds to geosmin, nothing else. And it responds very nicely with a strong, it's a small cell here, when it gets a low concentration of geosmin blown over it. Here is the in situ of these cells. So there is about 25, 30 of those cells on the antenna. Okay, so we established this one line. One cell is detecting this compound. So these neurons expressing these two receptors, or this receptor, OR56A, are responsible for geosmin, right? One problem there is that we didn't know that it was OR56A because this is one of the few neurons that by some reason expresses two receptors. And Peter Mombars has been talking a lot about receptors, I'm sure. Insect receptors are functioning more or less the same. I will not go into that today. But this neuron has two receptors, so we couldn't know which one is the active one. So normally then you take these receptors and you put them into something called the empty neuron system in Drosophila. There is a special mutant that lacks receptor in one neuron, and you take the receptor you're looking at and put it into this neuron and you can establish its physiology. By some reason this receptor doesn't work there, so we had to figure out something else. So now you get the whole strategy and how this project unwinded, right? So we had to resort to something else. What do you take? Yeah, we take Chinese hamster ovary cells, of course. So we went to the lab, we took the Chinese hamster ovary cells, we put our receptor into those cells, and then we did imaging of these cells and saw what do they respond to. And lo and behold, they respond very nicely to geosmin when we insert the 56A. When we insert the 33, no response to geosmin. So there we had established that 56A is really the geosmin receptor. Okay? So we're part of the way. So of course, if we want to sell this to somewhere good, we have to show it behaviorally as well. So if 56A really is the sole input, then if we silence it, we should also abolish the repulsion to geosmin, right? So we went in and we used a system called Shibiri. Shibiri is a very nice system because if you insert this dynamin system into the fly, into these neurons, when you increase the temperature from 25 to 32, you stop synaptic transmission. So it's a reversible system, so you can have your fly totally normal with 25, then you increase temperature to 32, then you stop the function, the transmission of exactly this message, nothing else, only the message regarding geosmin. And then you can go back to 25 and the fly is totally normal. So it's really a perfect experimental setup, right? So we did this in our system. We did 25, we did 32, and very nicely all repulsion was stopped when we shut down this nerve line. So it's up here. See, 32, when it's 25, very nice repulsion. With both the control lines, repulsion as normal, only when we activate the Shibiri system by increasing the temperature to 32. So, good. We are on the way. We have now shown in which cells it's detected, which receptor is responsible. We have a good behavioral response that is shut down when we shut down these cells. We did another control. We took a receptor for a very attractive odor that we know is extremely attractive, and that's 22A. 22A is another, it's a fruit, more or less, a fruity odor. But we express that receptor in the same neuron. So now when we blew that odor over the fly, we of course we activated the normal attractive line, but we also activated the geosmin line at the same time. And then we stopped all attraction to this very nice odor. So again, the nice odor started smelling both good, but also this stop odor. So all attraction abolished. Another good thing. Okay, so how specific and sensitive are these then? Is this a dedicated line, or is it just something that will detect anything that smells bad? Because you could think that it's economic for an olfactory system to just have one line saying this is bad, stay away. So again, we went back, singles and xylem recordings, started with 120 compounds that we know are good or bad in some way for us or for the flies or for someone else, and amazing specificity. Out of this 120 odors, only geosmin elicited a significant response, or a response overall. You see here, all of them are listed there. Only geosmin. We did imaging, we get response only in one glomerulus, the DA2, and that we know is the target of the OR56A expressing cells. So very specific response inside the brain as well. But of course, if you think about outer universe, how many molecules are there, right? 120 molecules, it's a piss in the Nile, right? So we said we have to really go in and look more from in a more comprehensive way. So then we resorted to a method that we like a lot, which is called GCSSR. That's when we link up a gas chromatograph, chemical instrument, with neurobiology. So we hook up a fly in the outlet of the gas chromatograph. So we can take any kind of odor. You know, the odor of a banana is about 900 molecules, right? So we can take, for instance, the odor of the banana, inject it into our gas chromatograph. The gas chromatograph splits this odor up into its 900 components and blows them one after the other over the antenna. And in this way we get an immense resolution of what the fly smells overall, right? So this is what we did, and we took anything that we thought could be relevant for a fly. We took the fruits up there. We also took bad things that should be like rhino shit or lion shit, hyena shit. We took good stuff like vinegar, and then we took some other sources. And the only time we saw a response was here, here, and here. And these peaks are all geosmin, because these are geosmin-emitting substrates. It's the only time. So here we tested over 3,000 compounds. And we think that we covered more or less olfactory space for the fly. And only geosmin elicits a response. So it's an amazingly specific channel. A real labeled line. So how sensitive are they? They are immensely sensitive, as I said before. They go down to concentrations that we have only encountered in a few systems before, like pheromone system in the moth, for instance. Okay, next level. Now we have looked at the nose. What happens then? So we wanted to know what goes on when the message has left the nose and it gets into the brain and the next level. So does the information then diffuse, or does it stay in this one line that we talked about from the beginning? So here's the brain. One Japanese postdoc spent 3-4 years recording from projection neurons going out from the antennal lobe up into the higher areas of the brain. Here is one, it's a bit faint here. Here's the one glomerulus neuron going up. And what we could see there was that he could get one of these neurons, one neuron, in 3 years, that invaded this glomerulus and it responded nicely to geosmin. Was it then specific? Yes, only geosmin. So the specificity of this system is preserved. We could, of course, also look in imaging by expressing the imaging now in the projection neuron, not in the receptor neurons coming in. And again, exactly the same pattern. You see here, only one glomerulus lighting up after geosmin stimulation. So we submitted to Cell, and they said, we cannot accept an N of 1, right? Cell cannot publish anything with one neuron. So at that time, Yuichi had got his position in Japan. He had left the lab, so I had to call him up and say, Yuichi, we're deep shit here. We need another Cell. So he flew back from Japan and spent 3 weeks, day and night in the lab, only looking for geosmin cell. Only day and night, and he managed to find one more. So we got an N of 2. Okay, so we wrap it up so far. It's really both selectivity and sensitivity here is really remarkable. It's something that we have never seen in Drosophila, and before we have only seen something like that in the moth sex pheromone system, where the male is looking for the female, and you know sometimes the male builds this amazing antenna and so on. We don't see that kind of adaptations here, but still the system is so sensitive. So it's really an amazing adaptation as far as we see it. So then of course comes the question, why? Why should you have a system like this? Why should you build such a sensitive detection system for particularly geosmin? Does it have anything to do with these guys, with the microorganisms that are emitting these compounds? So the first thing we did then was that we took flies and then we reared them on normal medium and we reared them on medium where we had put these microorganisms into it, one and the other. And what do we see? It's toxic to the flies. And even worse, penicillium. And these are typical organisms that will come in after something has been good for the fly. So you have an orange at home, you leave it lying. After a while, yeast will infect the orange. And then it's optimal for the fly. Then it's absolutely best, just when the yeast has come in and started growing. But then after a while, if you leave it, and I'm sure you know it, it turns green. And this green also smells bad to us. Often you can smell it when you get home and an orange or a lemon has turned green in your fruit bowl, you can smell it. And that becomes toxic to the flies. And they should really stay away from there, right? So this was still some kind of hypothesis that this is bad stuff and you really shouldn't oviposit there because your larvae will die. Both you and your larvae are harmed by this. So, of course, we tested oviposition. We went in there, we offered the flies to lay their eggs in normal medium or in a medium with these microorganisms growing on it. And there was a very strong preference away. They didn't lay a single egg, more or less, on the microorganism side. Then, luckily, in the lab, we had some microorganism working scientists because our question was, of course, is it geosmin? It could be something else that smells bad. So luckily we had some guys that worked on a strain of Streptomyces that is totally normal, but it lacks the enzyme to produce geosmin. So it's just as bad for the flies. It lives totally normally, it looks normal, but it doesn't smell of geosmin. And what happens? Puff. All repulsion is gone. The flies go there, they lay their eggs, and they die. So geosmin is necessary and sufficient for this thing to happen, right? So we could dissect the system from both ways. Of course, the same thing happens when you just offer them geosmin odor. They also stay away from ovipositing. If you do the same with the previous best negative odor, even with pure odor, you don't get any behavior at all, more or less. So geosmin is really unique. How conserved is this? So we are also evolutionary neuroethologists, of course. So we want to know, is this something that every fly would use? So we went into more or less every fly species that is in this group. There it comes. And very nicely, more or less every fly that we looked at has this kind of system, this specificity. Geosmin seems to be something in general that a Drosophilid fly should stay away from, irrespective if they live in the Sonoran Desert or in your kitchen or in Indonesia. Only one fly, we couldn't detect this at all, and this fly has evolved to spend its whole life inside a flower. So it's really never exposed to this kind of dangers as the other ones are when it comes to geosmin. So probably this gene, this receptor has pseudogenized in this fly and it's not expressed at all anymore. So in short, this is a sign of bad food sources. You should stay away from there. That's what this tells the fly, right? So presence of geosmin prevents attraction, it prevents oviposition, so it's something that is really bad for the fly. So we want to summarize this from a neuroethological perspective. This is what, as I said before, this is what the fly wants. Some fruit infected by yeast. This is what the fly doesn't want. The fruit with yeast, but now also with the new toxic microorganisms. And that's where they should stay away from. And we're extremely happy about this because it's really neuroethology in action. It's exactly what we want to do in my lab. If you want to read more about it, we got it into Cell late last year. We got the cover, and there is even a movie in the web where you can see me and Marcus Stensmer drinking wine in my kitchen, telling the story about this one. So very happy about this paper, I must say. Okay, So now we looked at bad stuff. So how do they code good stuff? Is this label line thing something specific when you have something bad, or would it also be when things are good? And everything I will talk about from now on is sort of a building site. So you will not get this kind of complete story as you got for the first one. So a fly has to deal with a lot of stimuli, right? It's a constant smorgasbord of stuff out there that could basically be good to eat. So it has to have some kind of preference. It has to have a preference to be attracted, where to lay its egg, where to feed, and so on. Are these things the same, or are they different things to the flies? It's different things to be attracted and then to lay your eggs and to start feeding, or different kinds of stimuli interacting, and so on and so forth. A lot of papers have been published on yeast. We had one from Alnap in the Linnaeus program, showing that yeast is really what the flies like a lot. The yeast more or less overrides any kind of other stimuli because it's really what they want to eat. So we started looking at yeast, and if you isolate yeast from flies, because when they carry this yeast with them all the time, you can find these different kinds of yeast species growing in their stomach or on their body. So we started looking at these different ones. And we let the flies choose, both when it comes to attraction, oviposition, and feeding. And they like several kinds, but one that almost always ends up on top is this Brettanomyces yeast. Brettanomyces yeast is what we use to make wine. So flies really like wine yeast. So the question is why then, again. So which odors could be involved in yeast choice overall? We started looking at Brettanomyces, and it turns out that it produces what we call spicy odors. Spicy odors, when you drink a good wine, often there is some kind of, that gets into your nose, right? The retronasal experience of a good wine with some phenolics, it's a full-bodied wine, sometimes we say, right? And it's often dependent on these kinds of odors. That's phenols, guaiacol, and so on. So these are specific to this kind of yeast. What is also nice is that they're not in the yeast per se. The yeast has to grow on a substrate containing these kinds of compounds, then it can produce the spicy odors. So if you think about this gives us, as ethologists, again, some possibilities, because we can manipulate the presence of these compounds in the yeast and see if it means something to the fly. We can cut out the substrate, then the yeast will not produce the spicy compounds, and we can see what happens, right? First we had to see, can the fly smell these compounds overall? Too big picture there. So we went back to the lab, we went back to the GC single sensillum recordings, hooking up the gas chromatograph. This should be swapped, that's wrong. This is without spice, so this is where we grow the yeast without the precursor. This is when we grow it with the precursor. And what we see here is that something happens down here. This response in one, now we're on the palp, so we're on the other olfactory organ of the fly. And we see a nice response from a receptor that is called OR71A, and it's expressed in a specific neuron. Okay? This is what it looks like. Here it is in this kind of sensillum. Very nice response to 4-ethylguaiacol. But if we compare it to the geosmin channel, here you see we have quite a wide response. But all of these responses are to spicy compounds of different kinds. Eugenol. Eugenol is the smell of cloves. The spice, for instance. Okay? So then we went in and looked at turning off 71A again. We used the Shibiri system again, and we could show that when we turn off this cell, we lose all attraction to the Brettanomyces yeast. So these are key compounds for the fly. We were guessing right. The spicy compounds is something that the fly really searches out when it looks for good yeast. So good thing again happened. Then of course we have other possibilities, and this is something that you might be interested in as well. You can not only turn off something, but you can also turn it on. So you can express heat-sensitive channels in this neuron that are excited by heat. So we express the TRP channel in the same neuron, And then we let the fly choose between low temperature and high temperature. So as soon as the fly goes into high temperature, it will activate the smell of good yeast. When it goes to the cold, it will not smell the good yeast anymore. So, of course, the flies all the time chose to stay in the nice warm environment where it was smelling of good yeast. So when they themselves activated their neurons by going to a warm place. So good result again. So why? What is happening here? Now, if we look at red wine, we're now more, these days we're told you should drink at least a glass, maybe 2 glasses, maybe half a bottle a day, because it's so good for you, right? It's full of antioxidants. And our question was, of course, could it be the same for the fly? Could these compounds act as antioxidants and therefore the fly go and eat Brettanomyces yeast? So we got hold of a superoxidant, paraquat. It's a terrible toxic substance that for a long time was used, sprayed in nature as an herbicide, but it turned out that it's really one of the worst oxidants creating free radicals you can think of. There are bad stories in the web of people who drank this, and you can read there yourself. I don't want to put the pictures on the web here or on the screen, but it's really bad stuff. Okay, but the thing that paraquat does is that it oxidizes very, very strongly in the cells of your body. So we took this compound, and of course we had to test it. So first we let flies live on yeast with these spicy compounds, the black line. We let them live on yeast alone, we let them live on glucose. They survived pretty well on all of these substrates. Then we gave them paraquat, and of course it was bad to them. So they died pretty fast. We gave them yeast with paraquat, we gave them glucose with paraquat. And then the final experiment, of course, we gave them this kind of stuff, but we added our spicy compounds. And that saved them to a certain degree. So it ameliorates the effects of paraquat, of the oxidizing. So there seems to be an antioxidant effect of these spicy compounds to the fly, and that might be why they are going for this kind of yeast, because it's healthy for them and it helps them. We haven't seen that they can smell the paraquat, no. So that's where we stand in this project right now. As you see, we haven't progressed so far, but it's still a cool system. One system where we have progressed a little bit longer is to look at a line for another behavior. So again, we think about the smorgasbord for the flies, and they are choosing between things smelling of microorganisms, things smelling of fruit. They are swarming over the table. But this time we wanted to look at how do they choose fruit? Because we know fruit is important. Even though yeast has to come in sooner or later, they always choose some kind of fruit to lay their eggs on. So what we asked here is, do they have an innate preference for certain fruit? Fruit in any sense, how we think about fruit. So we started letting them choose. And what showed up is that they really love citrus fruit. And the most they love oranges. Orange is the overriding choice. Every time, whatever you let them choose among, they will always choose the orange, as you see in all these experiments here. Tons of behavioral experiments. The peel has to be there. If you take away the peel, the orange goes totally non-interesting. So they really love the peel of the orange. Otherwise, they will not go to it. Okay, so it's great with citrus fruit. Good thing when we're in Spain, right? So what is typical of a citrus smell? Because obviously the flies really love to go to the odor of citrus. So as you see, there is one green peak popping up in every grass chromatogram of all these odors of all these different citrus fruit, and it's limonene. Limonene makes, in some of these, up 97% of the volatiles that are emitted by a citrus fruit. So we got very interested in this compound. We did bioassays. We could show that they love to lay their eggs where it smells limonene. It's a very strong oviposition elicitor. But interestingly, it doesn't elicit any attraction at all. So this compound doesn't seem to be involved in attraction to come to a substrate, but once you're there and it smells of this, you will lay your eggs. So these two lines are separated in some way. Luckily, we got hold of some Spanish people who had, by some reason, made a transgenic orange. And this transgenic orange they had made so that they had cut out the enzyme to make limonene. So here you have, again, an orange that is totally normal, but there is no limonene in it anymore. So of course we did the experiment. The flies will lay their eggs almost only on the orange that smells of limonene. The other one is totally normal, but it just doesn't smell of limonene anymore. And you see it comes down here to all the other fruit in attraction or in oviposition index. But again, there is no difference in being attracted to this thing, but you will just not lay your eggs on the one that doesn't smell of limonene. So limonene seems to be a very important signal to lay your eggs. Okay, so then we have a behavioral background again. What do? We do our things again. So where is limonene detected? Again, going back to the 4 different kinds of OSN, talking to Honey, my Egyptian superhero, and asking him, can you do another 10,000 recordings? Sure, he says, and then he records, and we find 2 main channels in that detect, and this one here is really minor. This is the main channel in, and interestingly, this is one where nothing was known. It was a mysterious sensillum before, a mysterious cell, because no one had ever found something that excited it. It's very sensitive to limonene. It has been mentioned in the literature for a long time as a mystery of Drosophila. So we looked at this, very nice response to limonene, but again, a wide response spectrum, not like geosmin. Here we have response to a number of compounds, but again, they're all related. They're all part of a citrus odor. They're all terpenes. So if you smell an orange, these are the molecules that you will get in your nose. And here we have one neuron that seems to be detecting all of these different kinds of compounds. The best one was valencine, named after Valencia, because of the oranges, right? It's one of the major compounds of orange odor, and that's the one that activated this neuron the most. But you see, there is a whole host of odors that activate it very well. When we gave the fly all these 15 best ligands for this cell, they all elicited oviposition. So whichever compound excites this neuron will make the fly lay its eggs. None of them will elicit attraction. So again, coming back to the same thing, it's not something that makes you go to where you should lay your eggs, but once you're there, this thing makes you lay your eggs. So then we went in and we did GCSSR on this kind of this neuron, and you see that every citrus fruit elicits very nice responses. So here is the limonene, here are all the other terpenes. In every one of these, we have these kind of responses. If we compare that to the other fruits that they don't like so much, this cell is never activated. In Sweden, these flies are called banana flies, but they're definitely not banana flies because banana odor doesn't excite this cell at all, and you see, of all the other compounds, not a single response. So it's really a citrus detector. Which glomerulus is then activated? We went in again with our imaging stuff. You can see it's a single glomerulus that is activated by this odor, and this is the DC1. We could see that, again, unluckily, it's a neuron that expresses 2 different receptors, 2 receptors that have diverged very recently probably. But here we were lucky, so we could put it into the empty neuron of the fly and dissect it in this way, and we could show that this is the 19A, sorry, oops, going too fast, the 19A receptor that is expressed in one of these neurons in that sensillum. And again, we could shut it down. We could show that if you shut this down with shibiri, you stop egg laying, and so forth, using the same methodology. If you let the fly choose between orange and plum, all the flies will lay their eggs on orange, right? So here again, we could test it. We shut the system down, and when we shut it down, this whole preference between orange and plum is gone. So this whole preference seems to be built on input from these neurons. If we do the same thing in the other way, so we add valencine to the plum, then also we lose the preference. So we can dissect it from both ways, sort of. Okay, there are also other OSNs, other neurons that respond to peel compounds, mainly two of them. It's these two down here, and we know who they are, we know the receptors, so we could test them. We could do behavior for these, we could do physiology for these. They respond to other peel compounds. So citral, nerol, for instance, farnesol, extremely specific. When we check these, they also elicit oviposition, but they do not replace, they do not make the flies choose the plum or neutralize the attractivity of the orange, sort of. So it seems like the limonene is really the crucial factor that makes them go and oviposit. Here again, how did things evolve here? Because we had the geosmin before where it was the same in every fly, more or less, right? So we had to think about how did this come about? Because if you're smart and if you know your natural history, you know that orange is an apple from China, right? It's a Chinese, it's an Asian fruit. The fly is African. So, why should these flies go for orange? How did this thing evolve? So you could think in different ways, right? You could think in one way that there was, in the very, very far back, we know that all Drosophilids probably came from Asia, and there was a grandma, melanogaster, who was sitting back in Asia, was exposed to oranges in those days, got to love them, then got over to Africa, discovered some fruit that had some kind of similar odor spectrum which maintained this kind of lines, and in this way we today can see that they're attracted to oranges. That's one possibility. The other possibility is that this never happened, but that Drosophila really is predisposed to find another fruit, but this fruit is really smelling similar to an orange, and that would predispose them to find the orange in your kitchen. Okay, so we could look at these different systems. And again, we went in, we did tons of GC singles and psyllium recordings again in the lab. We got colleagues to send us about 100 different fruits from Africa, fruits that I never saw. I didn't know they existed, like baseball large fruits hanging from trees, amazing variety of fruit. And we collected orders from them all, we tested them in the lab, and we found one. We found the African squirrel nutmeg, Monodora species, which elicits a very similar response from this neuron as compared to an orange. If we do a PCA of the molecular spectrum that it emits, it falls out here with the citrus, not down here with all the other fruit. And if we do the experiment, bam, there is no choice. The flies like the Monodora, the squirrel nutmeg, as much as they like an orange. It's around where Drosophila is found, about the same area. It's very abundant. So as far as we see it, this is a good candidate where the flies could be pre-adapted to really like oranges. So very good for us. Why? Again, before we had antioxidants, right? That might be something. Before we had geosmin, staying away from the microorganisms. So why should flies like citrus fruit? So we started thinking, what could be involved here? Well, parasitoids. 80% of larvae are parasitized in nature of Drosophila. So it's a very major selection pressure. So we started looking at the parasites. We let the parasites choose among Drosophila larvae that were living in oranges and in plums, and all of them went to the plum. We let parasites choose between citrus odors and a control. They always went away from the citrus odor, especially from limonene or balancine. So there seems to be something here, right? Then we tested the final experiment. What is the parasitation rate of Drosophila larvae if you let there be an odor present? And if you look at here, this is the solvent control. This is the parasitation rate when you let only the odor of a single odor of orange be there, and the parasites go, almost all of them, to the larvae living where it doesn't smell of orange. So you take away an extremely strong negative selection pressure if you let the larva live on an orange or where it smells of orange. So again, a nice correlate between how the sensory system has evolved and what is happening out in nature. So what is happening in other species? Here we have another situation, because before I showed you that geosmin is probably important for everyone, right? Here you have to have a neuron that makes you lay your eggs in the right site, in an optimal site. And here every species seems to have adapted this neuron to where they live. So everyone has their own response spectrum. And if you look at this neuron, it's the one that has the second strongest positive selection of all receptors in Drosophila. So there is a very strong selection pressure on this receptor to change, to be plastic, to adapt to where you live. You know, there are Drosophila species that live in vampire shit. That's where their larvae live. There are Drosophilids that live in rotting potato. There are Drosophilids that live more or less anywhere. And of course, they are all adapting this neuron, because this is the neuron that feeds into the oviposition circuit. And you have to adapt it all the time to lay your eggs where you should be, right? The vampire guano fly shouldn't lay its eggs on an orange. It wouldn't be good, because the larvae wouldn't survive. They need to be in the feces of a blood-feeding organism, right? And the other way around. So you need to adapt your system all the way to fit in where you should be. So briefly, this is wrapping this whole part up a bit. So we found a very nice correlation between a single line through the system and the behavior of the fly. So then, we still have some time. In the second part now, the last 15 minutes, I want to go into more neurobiology, more circuitry, more higher coding in the brain, and look at how is valence coded at the next level of the brain. So this is what we looked at before, the antennal lobe, and then we have these pathways going out of the antennal lobe, hitting the mushroom body, hitting the lateral horn. Some more. We were looking at this, and a lot of people have spent a lot of time on this pathway here. It's 80% of the output, I would say, of the antennal lobe. It goes to the mushroom body, it goes to the lateral horn. It passes by the plasticity center of the brain, then it goes out to the lateral horn. But then we have this little one here, and it totally bypasses the mushroom body, and it hits the innate, what we think is the innate response center of the brain directly. It bypasses all the memory stuff, we think. So we were interested in this part, and we wanted to dissect it. So this is what these neurons could look like. This is the normal pathway. So one neuron here sending its axon out, branching in both of them. So luckily there is a line. There is a GAL4 line of the flies that is specific for these neurons. So you can express a dye in them, you can see them very nicely. This is the normal ones. These are the ones bypassing the mushroom body and going directly to the lateral horn. You see it here in a schematic way. Okay, so we wanted to look at these neurons. What do they do? The first thing we did was to look in the antennal lobe. How do they innervate if you compare it to the other neurons? And then you see that they have quite different innervation patterns. They are very different in other ways as well. So the ones moving through the normal pathway, the big one, they have uniglomerular innervation. So they pick up all their information more or less from one glomerulus. These ones, the green ones, as you see up here, they have very large innervation in the antennal lobe. So they pick up their information from a lot of different glomeruli. Makes it much more complicated, because for the other ones you could think that, okay, they get, like we saw for the geosmin, one odor coming in, one odor going out. That's not the case here. They are picking up information, they are integrating information from a lot of different kinds of input. Okay, so here is a single one of these. So by using photoactivated GFP, this is that when you can express GFP in these neurons, but it's not activated until you blow your laser onto the cell body, for instance, of this neuron. So here we directed our 2-photon laser onto this single cell body here, and you see that very nicely one neuron lights up. It's a very powerful technique to dissect single neurons in the brain of Drosophila, and then you can register them into a model brain in this way. We could also do imaging out in the lateral horn. So we expressed calcium-sensitive dye in these neurons, and then we looked out in the lateral horn what happens when you activate these kinds of these neurons with different kinds of odors. We could test good odors, we could test bad odors, and we could look at the activation pattern out there. And what we saw was that we could really observe very specific activation areas. We could see activation in a green area, like up here. We could see activation in a blue area and in a red area. And these were more or less not mixed up. So depending on which odor we blew onto the antenna, we saw neurons feeding into different parts of this, what we think is an innate behavioral center out there. And then we could connect this to attractive, not attractive, or bad odors, and also to concentration in some ways. And what we could see was that in one area we only had activation by good odors. In another one we only had activation by good odors. Bad odors, good odors. In another one we saw nice concentration coding. So there seem to be different modules in this lateral horn that take care of different kinds of information. So then we had to look at, is all the activity that we see really contributed by these neurons, because this line is not pure. There are also some other neurons that are marked when you go in and work with the genetics behind this line that we're working on. So then we went in and we did photoablation, because we know that there was one neural line, or in this line neurons coming in from here, feeding in up to the lateral horn, were also marked. So we did photoablation with our laser, cutting off this line or cutting off this line, and seeing what happens to our activation patterns when we take out some parts here. And then we could see that more or less one area, the red one from before, was totally dependent on the higher order neurons going out from the lateral horn, while the other two, the green and the blue areas, were dependent on the input from the antennal lobe. So the lateral horn has input into two areas, and then it's giving output from one area in this, what we are looking at here. So this is what it looks like. So when we lesioned what was coming in from the antennal lobe, then we only got activity down here. When we lesioned what was going out of the lateral horn, then we only got activity up here. So blue and green antennal lobe input, red lateral horn output. Okay? So we could really separate our neurons into different classes, innovating these different parts. So sticking to the colors, green neurons, blue neurons, and we could connect this to the activity that we saw from different kinds of odors that I was talking about before. Good odors, bad odors. So the green ones, bad odors, the red ones, good odors, and here some kind of concentration coding. Again, combining all of these into the general atlas of what we were seeing. So these neurons are negative, they are inhibitory, they're expressing GABA. So what we could do was we could shut down this line of information going from the antennal lobe by expressing RNAI against GABA synthesis in these neurons. Then you shut down the activity of what they're doing, and we could observe what kind of effect do these neurons have. And what's very interesting for us is that when we shut them down, everything got much worse for the fly. Here is behavior. What is most interesting is out here, because that's what happens with every odor when we shut down this line, right? These are two different concentrations of the odor, high concentration, low concentration, and when you see movement to the left, that means that the fly gets turned off. So when we shut down this information flow, good odors became neutral, more or less. Neutral odors became bad. Bad odors become very bad. Very bad. The only thing that stays the same is very bad odors, because they cannot get much worse, right? Like acetophenone here, not much happens because it was so bad already from the beginning. But so it seems like the major pathway from the antennal lobe is telling bad stuff, and that this pathway is inhibiting that bad information and allowing positive chemotaxis towards odors. So when we do the statistics of this, we could see a very strong valence correlation in the green area for bad stuff, a very strong valence correlation in the red area for good stuff, and here correlation both for bad valence and for concentration. So different kinds of coding in these different kinds of areas. This is what we see in the antennal lobe. So not much coding of valence as when you compare to here in the lateral horn, where there is a very strong separation of what is good and what is bad in the higher areas of the brain. Okay, so wrapping it all up, first we looked at these 110 odors where we could show that the antennal lobe is sort of separating the message to some extent in its output. Then we looked at geosmin, this dedicated pathway for a bad odor, telling the flies to stay away from there. Then we looked at these antioxidant odors that seems to be the odors responsible for a fly going and choosing a specific kind of yeast, the wine yeast, Brettanomyces. And in the fourth project, we looked at the oviposition pathway, where the fly seems to prefer citrus fruit, and one of the reasons there might be that citrus protects them from their enemies. Sort of the enemies really stay away from citrus. And then in the last part, we looked at how we are looking at a pathway going to higher areas in the brain, and in this way trying to understand how are different information lines going from one primary sensory center up to other centers, how do they interact with each other to build a final kind of behavior where we can, and there we could see that it seems like one is feeding negative, the other one is inhibiting this negative inclination of the fly. So if you shut down the inhibition, the flies, everything becomes bad, more or less, for the flies. So as always, I should say who did a lot of this work. These are my group leaders that were involved. Marcus Knaden is the guy who did these 110 odors in the beginning. He's been involved in all of it. Silke also involved in all of it, doing the imaging. Marcus Stensmer, really good guy, where we worked with all the evolutionary ideas, geosmin, limonene, the yeast. He just got a job now in Lund in Sweden, moved up a month ago to a lectureship at Animal Physiology in Lund. And then, of course, this guy who provides us with a lot of money so that we don't have to write grants all the time. That's Max Planck. Our society is called the Max Planck Society after this famous German Nobel Prize winner. So I'm really working in an environment that is paradise for scientists, where we should do basic science. We shouldn't care about application. Our task is to deal with basic science and create results that other people can act on to make applications and so on. But we should stay with the basic science. This guy, he said, knowledge must precede application. So before you can make something, you have to know what it's all about, and that's what we're trying to do. This is my group. Out there with the red arrow, that's Hani, the Egyptian guy, who has just done an amazing number of recordings for all of this. And up there is our Japanese friend who had to come back and find another neuron. Very nice environment to work in. So that's it. Thank you.
Question?
Yeah, I'm curious with your oviposition pathway. I bet if you've done this, you would say it, but what happens in males? Do they have that receptor? Do they have that glomerulus that lights up? How do they respond to those odors?
Yeah, they have them, but we don't know. I cannot tell you what they do yet. We haven't had time. Of course, this is a question we ask as well. Why do the males have it?
Okay,
Because often.
I was expecting. So What's
Yeah.
your speculation?
My speculation is that the Drosophila often encounter each other for mating on the substrate. So it might be good for the males also to detect where it's good to lay an egg, is because it might be where the females hang out. And that's what we've seen for several others. That should be sort of female-specific odors, but where the males pick them up as well. And the same with the sex pheromone, right? The Drosophila sex pheromone is very strongly synergized by food odors, for instance. So they tend to meet. The meeting place is on the fruit,
But they're not attracted to.
no, they're not attracted to this one. So that's sort of contradictory to this. But that's the only idea I have in my head right now. And as I said, these are ongoing projects. So it's not a full story like geosmin yet. But any ideas are welcome.
I'll get back to you.
Yeah.
Two questions. In the lateral horn, what about interconnections between those different parts in terms of direct neuroanatomy?
Yeah, we know that they are connected in there, but no one knows. I mean, no one even knew what this line was about. So we have no idea how they interact with each other. But obviously there are connections going on between this input and this input, and if they are directly connected or if that is mediated by these more local circuits in there, we just don't know. We know they are there. But it's even in the antennal lobe, we have these huge neurons that connect everything, and we still don't know what they do. So I think local circuits is something that we still fail to understand to a very large degree. What do they do? Is it like contrast enhancement? Is it? No, we don't know.
Specifically, the ones you would expect the ones for concentration that to interact a lot with the others and so on.
Yeah.
Now, with the predators and avoiding where the parasitic wasps are, do you expect that there is or there might be some kind of arms race?
Oh, definitely. Yeah. So, that's going on all the time, and you bet that there is. So we're looking at the parasite now. We have gotten as far as we are doing singles and cilium recordings as we speak, and we found very nice cells in the parasitoid that detects these compounds. So obviously there is some relevance, otherwise it wouldn't have them. But of course, if all flies suddenly moved there, the parasitoid has to adjust in some way. But I think there might be enough flies on apples or whatever way you want to go, but that's a constant arms race. Yeah, cool.
Yes, I got one question.
Yep.
Something about the mixture of the good and bad colors. For example, when you got the lime and it starts being rotten, so they stop laying eggs on the rotten lime.
Yeah, when geosmin comes, there will be a total stop. But what we tried to do there in this one was to have fruit without. We tried to choose fruit where not even yeast had started growing, right? Because as soon as yeast started growing on the plum will get more attractive than the orange. Because yeast is always an overriding, and that will also come in the parasitoid story. I mean, so we have tried to dissect it from a minimalistic point of view. To start somewhere, we try to, because then we take the yeast per se and we take the fruit per se, and we show, okay, now we have very good yeast, they like it because of this. We have very nice fruit, they like it because of this. So maybe the optimal stimulus is an orange with the yeast, right? But then what happens? Then it becomes a super stimulus. But then you can have everything. Then you can have a plum with yeast against an orange with no yeast. You can have a plum with yeast and geosmin against you can get any kind of interactions. And what we know is really good for the fly is a fruit with yeast. That's the optimal thing for them, for survival. Because if you take a grape, is one of the other papers I showed. If you take a grape that is sterile, and you take a fly that is sterile, not sterile in the sexual sense, but it's not carrying any microorganisms on its own, put them together, the fly will die So the fly cannot, in the long run, make use of pure fruit stuff. It needs the yeast there to survive. That is what it's really eating. It can stay alive for a little while from the pure sugar, but after it will die much faster than if you have the yeast growing.
So the behavior of the fly is highly affected from what it smells, right?
Yeah.
So is there any influence, let's say, from the temporal properties of the algae? Like, for example, in the moss there's like an coating.
Yeah, exactly. It makes a difference for the fly as well, and that's what we have many other projects going on, and one is looking at the receptor structure, and one of the reasons why we think they have this strange receptor structure as they have is that they need to code these fast pulses, and we know that it's very relevant for the fly as well, that they smell fast. They can code very fast fluctuations in odor. And often also they are good at separating at a very, very fine scale if something really comes from the same source or not. Because it could be that there is one very good orange lying here, and then here lies an orange with geosmin. And those two odor plumes will travel downwind, but in physical sense, the filaments of the odor will never totally come together. They will always, like cigarette smoke. If you have one cigarette with pink odor and one with green odor, the smoke will travel downwind, and it will never be pinkish green really. It will be pink and green filaments traveling downwind. And that's what they seem to be extremely good at separating, that there is really one orange up there that hasn't geosmin, even though there is one that's smelling bad over there. And then they can choose to go. And that's what moths do as well. So they can separate the female, only the right female from the wrong female, in that the pheromones are not totally mixed up. Millisecond resolution.
Did you see any signs of centrifusion, meaning that perhaps vision and all would go together or connect to similar numerali, or?
In the antennal lobe, that's vision more or less. But higher up, when we're starting getting to this center that I talked about now last, vision definitely gets important. But there we're also looking at different species. So there is one invasive species now called Drosophila STOP that is invading both Europe and America. And it's very bad because it attacks fresh fruit. So it attacks cherry, it attacks strawberries, all these high-value crops. So it's a big thing in the EU right now to learn about Suzuki and what we're going to do against it. But the interesting thing is that Suzuki then goes, of course, for much earlier. Drosophila will not go until the fruit starts rotting, more or less. That's when they get really interested. But Suzuki goes just when the fruit turns pink. So we have worked on strawberry, and then we have done flower, green, white, whitish pink, pinkish white, reddish red, green, rotten, sort of, right? And in Suzuki, you turn on cells when it's starting to go pink. In Drosophila, other types of cell doesn't get turned on until it's more or less rotten. But Suzuki is also extremely sensitive to color. So for melanogaster, odor is totally overriding color. In the other one, color seems to be much more important, and they look for this red. When something starts turning a little bit red, that starts overriding the odor almost, because there is, in the strawberry field, there is now one strawberry over there that is turning red, so phew, I should go there. So there seem also to be different things going on in different kinds of flies when it comes to sensory fusion. They also like different kinds of colors. So Drosophila, I don't remember, but let's say Drosophila likes blue and Suzuki likes red. So they really have, if you put little balls of different color where they are trapped, they will go to different balls. Also, if you let them choose between the smelly thing and the colorful thing, all melanogaster will be in the smelly one. All Suzuki will be in the red one. So there are definitely things going up where you're fusing sensory input into one impression higher up, of course. But that also differs very strongly between pretty closely related species, totally dependent on their ecology.
I find that amazing how you were able to modify the behavior of the fly by genetic manipulation, would you say that the flies are completely pre-wired? Like there is no learning going on, no plastic, no synaptic?
Oh, there is lots going on, but we haven't let them learn, right? We are working with naive flies. So we are working with flies that come right out of the breeding station, let's say, right? Flies are extremely good learners. They learn after one or two experiences to associate with something good or with something bad. We know that. But you also know that there are some things that are so important. We were just discussing it before. Some things are so important that you shouldn't learn, right? Geosmin is probably so bad. It's a signal for something that is so bad that you should always stay away from it. You shouldn't learn. Just stay away. Forget about learning, right? I would think that the other channels might be much more prone to learning. Maybe if you're exposed to, if you wake up, if you're born in an apple field, like the image I showed you first, it might not be so optimal to fly around and look for an orange your whole life, because there is plenty of apple around and it's okay to lay your eggs there, right? It might not be the optimal from a parasitation point of view, but there are plenty of this resource. So of course there is tons of plasticity going on, and that's what's going on in the mushroom body part, where we know extremely little. So definitely a lot of sensory fusion, a lot of plasticity, and I have absolutely no idea what goes on there, because it becomes so complex, and that's why we try to stay further down line for now to establish these first. And only, as you said before, what happens when you mix these things up? You can only start playing with odors. Only mixing one odor and another one, I mean, in different proportion. What starts happening? How do they interact? And that's interesting enough. Yeah. More.
I really have two questions on how the odor receptor works. The first one is when a neuron is not selective for a compound, is that the receptor, is that it expresses several receptors, or that it's selected to a common part of the compound?
This is one receptor.
It's one
receptor, and that receptor has what we think. We still don't know how it works. No one knows how an odor is recognized, but we then think that this receptor has a cavity or a receiving structure that is flexible enough to recognize something that all these terpene molecules has in common, for instance, or all the spicy compound has in common. So for all of these, there is some kind of chemical theme for the ones that I showed you, but we also have receptors that seem to be able to respond to very different things. But when way back in 80s, I did a lot of work on pheromone receptors in moths, and then chemists did 75, 100 different permutations, and we probed the receptor cavity. Now the pheromone receptor is very specific, so it's more to compare to the geosmin, right? So the geosmin would be a good thing to start probing also too. But what we could show in those old days was that it's more or less molecular mechanics. So you can calculate how the neuron will respond from the energy that is needed to twist the molecule to fit into one low energy constellation, sort of, right? But then it all comes to how big is this cavity? Is it this big? Then maybe this many molecules will fit in there. Is it this big? And only geosmin. And geosmin is nice because you can do different enantiomers and stuff, but we haven't gone that way yet.
And the second one was when you were talking about the modulation of the pressure selectivity on OR19A.
Yeah.
You were saying that there's a lot of pressure selectivity pressure.
Yeah.
You were saying how does it work? I mean, when the Drosophila replicates, it is in a place where it gets mutated frequently, or it gets transposed, or what?
You can rank all receptors when you look at them in different species and so on. You can all rank them according to how they have evolved, and the faster you evolve, sort of, the stronger selective pressure there is, the more plastic you are to adapt. So the geosmin one is one of the most conserved. That's one that should be that is kept through all of these. But this 19A, as it is so plastic, that's why I say that there is a very strong selective pressure to adapt all the time, and that's where you take this conclusion from. But you can do all the bioinformatics as well, which I don't know how they do. But this is what this last picture was built on, that where you have calculated exactly how all the receptors fall on this scale, and that 19A is the second most fast evolving receptor in the whole gang there. But the specifics of how you do these calculations is not my cup of tea. Oh.
So, first of all, thank you for your fascinating talk. I think that next time we will meet the fly, we'll be more curious about it, rather than trying to kill it. So, I'm quite curious of what lessons can be learned from from a computational or robotic point of view. And so I have especially one question. So you mentioned that there are some receptors and that are very specific for odors, for attraction and ovulation. Mm. So it seems that some odors, so citron is more for ovulation, not so much for attraction. Yeah. You also said that when you look at the antenna lobe, you see some value coding at the output level for good and bad. Yeah. But do you also see separation for good for attraction, good for ovulation, and bad for attraction?
Yeah. Good point. So the one that we don't have many of them, right? We have one for oviposition. This one for oviposition seems to be in the middle. Geosmin falls among the bad ones, so that's good. The oviposition seems to lie somewhere in between. So we're not sure what's happening there. And that's the only example we have so far of something like this, right? So we have to look at the other ones. We now have the farnesol and the citral one. So we have several ones to look at. But I mean, as you saw, the areas that I showed, they only took up small parts of the antennal lobe. But where I think all this comes from is that the system tends to evolve from how should I say, receptor duplication. So you have one glomerulus here, and then this receptor duplicates on the antenna by some reason. So for instance, the one where we have 19A and 19B in the same neuron. You have the same among the new receptors, the IRs that Richard Benton works on. There you have you have 75 ABC. They're sitting on the gene, tak, 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 tak. So they are duplications of each other. And they often respond to similar stuff. So what seems to happen first is that there is a receptor duplication, and then in the long run in evolution, this triggers a separation of the glomeruli. And as the first one was a good odor, and this receptor changes a little bit, maybe the second one is also, there's a pretty good chance that that's a good odor as well, right? From pure chemistry. And that's how I think these clusters have built up. That maybe there was one good glomerulus, the first glomerulus, and that was lying here, and there was one bad lying there. Let's take a very simplistic view. And then from them, new glomeruli have been born, and these clusters have come about. But then I'm also sure that there has been totally new events, because we know that receptors are spread over the whole genome, right? So there has also been totally new event where a new receptor has come about, and that glomerulus is lying somewhere totally different. So I doubt that there is a real functional significance of having clusters. I more think that it's a developmental evolutionary consequence that you find these clusters of good and bad.
Well, from a more functional point of view, so if this line has to express, let's say, two different behaviors. Yeah. So one question from a more robotic perspective is at what. So if this separation, you can also find it very early, already in the sensory processing, in the value representation, or if everything is processed in the same way, and then only at some point you have two different behaviors. So it seems from your evidence that already the sensory coding
already out there. Yeah.
And then also the valence coding, I would expect, well, not sure of that, but could be guessed that you would separate the valence for one behavior. Yeah. So something can be good for one behavior but neutral for the other behavior.
But it depends on how you decide to build your system as well, right? Because so I've been working with Paul for 12 years now. Do you decide to build your system on labeled lines? Are you only building a robot that is going to roll out into the minefield and smell that, here it smells of cyclohexanone, all right, or TNT? I mark it. Then you only need one receptor, more or less, right? That tells this. But if you're going to build a smart robot that is going to be able to smell a lot of different things, then you might need to resort to this much more elaborate cross-fiber coding where you can have 10 different receptors. I mean, we can talk forever, but we are working on crustaceans as well. So we're doing quite a lot of work on Christmas Island on giant crabs, land crabs that weigh 5 kilos, and we're interested in them because they have evolved land smelling in 5 million years. They've only been on land for 5 million years, so it's like that, right? And our impression of them now, because we have done the transcriptomics of their antenna, and we only find the ancient type of receptors, which are called IRs, which are very, very old receptors related to our neuroreceptors and so on. And we only find 10 or 15 of them, right? The fly has 60, we have 350, a mouse has 1,000. We only find 10 or 15, but at the same time they seem to have the most astute, acute, sensitive olfactory system of almost any organism we have seen. They're amazing at smelling their way around. You put out a little drop of something and just like that there are 5 of these crabs there trying to get at it, right? And what we see there is that what our impression is that the fly has built a system like this. So you have 60, 70 receptors. They go together into an antenna lobe, where it's not so much processing going on. The crustacean the land-living crustacean seems to have done something like this. So they put 15 receptors out there. But in here, when you look at the marine living relative, who doesn't seem to be so odor dependent, he has one cluster of glomeruli, right? Then the big ones living on land, they have added so half their brain is now olfactory tissue. And when an insect has a few hundred neurons handling information going on, they have a few 100,000 neurons shuffling information inside the primary center of the olfactory system. So there again comes back to your question, should you invest a lot out there, or should you invest a lot in here to make sense of your environment? And our impression is that the fly invests a lot out there, and the crab invests a lot in here. And that's really cool to look at that brain, because I can give you any reference, we have published a lot on it now, but that brain is amazing. And then that has taken so much space, so their mushroom bodies, these guys live 100 years. Amazing organisms. Their mushroom bodies, which in crabs are called hemiellipsoid bodies, the brain is taking so much space now, so they have taken their mushroom bodies and shoved them up into their eyes. So the projection it goes from the nose into the antennal lobe, but then it projects up there into an amazing structure that looks so much like a mushroom body, but much, much more elaborate. And we know that these guys, they are smart. They will not play with you like an insect. I mean, if you only pick them up, they say, fuck off, I'm just going to sit in this corner for a week now and I will not play with you. So they are so much more smart than an insect is. So I think so much more is going on there. So I think there are very different ways that evolution can play with the system to let it do what it does. And that's why I think it's also very important I've discussed it with Paul a lot, also for robotics, to think about this, to look at what has nature has made experiments for millions of years, right? So to look at these different systems and how do you find an optimal solution for this? If you're only going to look for one odor, fine, and just a labeled line, right? It's like the geosmin system. But if you want a versatile system that is highly sensitive, smelling many odors, then you should go out there and read up on different organisms and say, maybe we want to do it like a fish, or we want to do it like a crustacean or like an insect. I think that there is a lot to learn. Yeah.
Yeah, I think we could carry on, obviously, but we should have coffee, and then we have another great talk starting at 11:30. So let's say thanks again to Phil for